Welcome to this video on translation. This is your cell and molecular biology course, and I'm your person step revenue control. In this video, we will walk through how a cell reads its genetic code to produce proteins, a life-sustaining process called translation. Translation begins once an mRNA transcript exits the nucleus and enters the cytoplasm. The ribosome binds to the mRNA and scans for a start codon, which is usually AUG, where translation officially begins. Now let's meet the main players. The ribosome acts as the factory. The mRNA carries the coded instructions in the form of codons. Transfer RNAs or tRNAs deliver amino acids, and amino acids are the building blocks that will form the final protein. So these are the key players in translation. Now let's see the process of translation. Again, like transcription, it involves three steps: initiation, elongation, and then termination. In initiation, the small subunit of the ribosome binds to the mRNA. A tRNA carrying the amino acid methionine binds to the start codon on the mRNA. Then the large ribosomal subunit locks into place, forming a complete initiation complex. In the illustration, you can see the initiation step of translation. You can see the small ribosomal subunits and then the mRNA. And then at the start codon on the mRNA, you can see the initiator tRNA with methionine amino acid binding to the start codon. The start codon is AUG, and so the amino acid will be UAC. About the genetic code and the codons and for which amino acids they code for, we have seen in the video which is on central dogma of molecular biology. So AUG codes for methionine, and followed by that with the use of GTP, the large ribosomal subunit joins, thus forming an initiation complex. The next step to initiation is elongation, which is reading the message. The ribosome now begins elongation, that is, reading the mRNA one codon at a time. Each matching tRNA brings the correct amino acid, which gets attached to the growing protein chain. In the illustration, you can see the elongation process in which you can see the incoming tRNA bringing the amino acid and then the exiting tRNA. And you can see the growing polypeptide chain where the amino acids are being added as per the information in the mRNA. So this is how the protein synthesis takes place, and this is the elongation step in translation. Every codon on the mRNA matches with a complementary anticodon on a tRNA. This ensures that the protein's amino acid sequence is built correctly according to the genetic blueprint. That is, according to the information which is present in the DNA, the mRNA is formed. That is, according to the information present in the DNA, the protein synthesis takes place. We have seen in the transcription that the mRNA formed is complementary to the DNA template. And then in the translation, you can see the anticodon found in the tRNA, which brings specific amino acid to the site. You can see it is complementary to the codon present in the mRNA. So this will ensure that the amino acid sequence is correctly built according to the genetic information present in the DNA. Peptide bond formation. As each new amino acid arrives, an enzyme inside the ribosome forms strong peptide bonds between them. This growing chain folds into a complex shape which is determined by its sequence. The enzyme involved is peptidyl transferase. In the illustration, you can see the mRNA and then the small and larger subunits of ribosome. And then you can see three sites on the ribosome, P, and A. A site is the site where the tRNA, which is entering the ribosome, attaches to, and then the P site. And then from the A site, it is translocated to the P site. And from the E site, it will exit the complex. And then the next tRNA arrives at the A site. And then it is later translocated to the P site. And between these two amino acid, which is brought by these tRNA, a bond is formed by the action of peptidyl transferase. This bond is called peptide bond. So with the formation of peptide bond, the amino acids form a sequence accordingly. After releasing the amino acid, the tRNA from the site P is transferred to E and then it exits the complex. You can see the peptide bond formation in the illustration. So hope it is clear now how the peptide bond is formed between the amino acids present in the complex of ribosome and mRNA. So we have seen that the tRNA moves from A site to P site. Now that process is called translocation. Once the bond forms, the ribosome shifts over by one codon, a movement called translocation. The empty tRNA exists and A new tRNA brings in the next amino acid, thus the cycle is continued. So in the illustration, you can see the moving of ribosomes from the A site to the P site, and then the peptide bond is formed. As the ribosome moves, the tRNA is transferred to that is, it is translocated to the P site, and then when it moves to the E site, it will exit. And later, upon another translocation, you can see the exit of tRNA, and then the tRNA with both amino acids are present in P, and then the cycle repeats, such that a chain of amino acids, according to the codons present in the mRNA, are formed. So the next step and the final step is the termination step. When the ribosome reaches the stop codon, usually there are three stop codons, which are UAA, UAG, and UGA. When the ribosome reaches any of this codon, which is the stop codon, no matching tRNA is available. Instead, they release the factors that bind to the ribosome and promote it to release the newly synthesized protein. In the illustration, you can see the release factor, which is on the stop codon, and then it will release the empty tRNA, and then the free polypeptide, which is the protein, is released, and then the translation complex is dispersed. So this is how the termination step takes place. The protein then folds into its final three-dimensional structure, which is often with the help of specialized proteins called chaperones. So after these modifications, now the protein is ready to perform critical tasks inside the cell. In the illustration, you can see the pyridine structure of protein, which is then from the alpha helix or the pleated sheet, which is beta sheet, and then it is the secondary protein structure. Then it forms the three-dimensional folding with the chain interactions, which is uh, which occurs according to the structure of the protein. Then finally, it forms the quaternary protein structure, which contains more than one amino acid chain. So this is how the protein folds into functional shape, and then it begins to perform the functions inside the cell. So translation is the remarkable process where genetic information becomes functional proteins, and ribosomes, tRNA, amino acids, and precise reading of mRNA all come together to sustain life at the cellular level. So with that, I hope you got an idea about the process of translation. So we have seen the transcription and then the translation, which are the steps involved in the central dogma of molecular biology. So with that, you have got an idea about how the information in the DNA is passed on to proteins, and then this protein carries out the functions in the cell. Thank you for watching. See you in the next video.